Hello! So here's our intro to our first big lab, is yeast alive? We're going to figure out if the yeast is a living organism or if it's not. And here's how. You're going to need 1 packet of yeast, 1 packet of sugar, 2 red balloons, 2 yellow balloons, and a 100 milliliter glass of water. In addition, you're going to need a test tube tray with 4 test tubes labeled with tape 1, 2, 3, and 4. You're also going to need a funnel and a stir rod. I've already begun so that it goes a little faster, but you're going to fill this test tube up until about halfway. Paper says 4/5. And then you're going to see I've spilled. We're going to avoid that by cutting this open with scissors and using a funnel. You'll dump half of your yeast packet into your first tube. Share the other half of your yeast packet with a neighboring team. Once you've poured half of it into the tube with the water, you're going to pour a little at a time, and then using the stir bar or your thumb over the top, you'll invert it or stir it. But be careful, if the water is to the top, that means once you put the bar into it, it'll overflow. So you want to make sure the water is not to the top. So I would only add about maybe 2/3 with water, pour your yeast in, and then stir it. After you've stirred it, you're going to take this funnel and put it into your other empty tubes. Take your first tube and dump a little bit in. You want to make sure that the mixture is cloudy like this one is, and each one of these, so you have the same amount. You'll have a little bit in the bottom of each. Then you get more water and add it so that all of them have yeast and water that comes up to about 4/5, almost to the top. This one's too full. Then what we're going to do is you're going to take red balloons. The red balloons we're going to put over test tube 1 and 2, and yellow balloons 3 and 4. But before we do that, your procedure says for test tubes 1 and 2, you're going to put a half a packet of sugar in each one. So let me put a half a packet in this one, half a packet in this one, maybe a little more here, a little more here, a little more here. Oh, it's a big packet. And then you're going to put the balloon over it. You want to make sure that your test tube's inside of your tray when you do this. And I'm going to put my balloon over it. I'll move this one out of the way, like so. Now the balloon's over it. It hasn't been mixed. The sugar hasn't been mixed. So I'm going to go ahead and put my thumb over it again. Invert it. Time or 2. So the sugar's been mixed. I'm going to do the red one on 1 and 2. That's with the sugar. The yellow one's on 3 and 4. That's without the sugar. Ignore the fact that that says 3. And then I'm going to sit and wait. So half of your team will sit and wait and watch the balloons. You're going to note any change in the balloons in your table every 5 minutes for 25 minutes. The other 2 team members will begin working on the second half, the reproduction. You'll find 1 petri dish with agar in the bottom. You'll still use these yeast packets, which aren't completely gone, and a few drops of water. What you're going to do is you're going to add a few of the yeast grains and then add drops of water on top and then recover the plate. Okay, enjoy.